So we're at now 3 minutes past 4, so I think it's probably a good idea to start. We'll do a welcome to country, and I'll introduce Jelte, and then we'll get underway. Okay, Jelte?
Right.
Excellent. So I'd like to start by acknowledging the traditional owners of the land on which we work and meet today. The Wurundjeri people of the Kulin Nation. Pay my respects to their elders past, present and emerging. And welcome all of you who are here. Thank you very much for coming to see Jelte's PhD completion seminar. Jelte is now back in the Netherlands, but he joined our lab in 2018, right at the end, so in November. So he effectively started his PhD experiments in 2019. And he left in January this year, so he's had just over 3 years here at WEHI with us. So this is a pretty good amount of time, but quite short if you're going to do a PhD on the liver stages, especially the liver stages of the human malaria parasite, Plasmodium falciparum. In 2015, Jelte did his Bachelor of Science, majoring in biopharmaceutical science at Leiden University in the Netherlands, which is a familiar place to many of us as malaria researchers. Then thereafter, he did his master's degree, and again, that was in biopharmaceutical sciences at Leiden University. And he did that with Chris Janse and Blandine Frankie Fayard, who are well-known malaria researchers. And in his master's degree, Jelte generated and characterised some Plasmodium uelii mutant sporozoites that could not glide properly, and he measured the way that they undergo in vivo infection using different routes of administration. And as part of his master's degree, Jelte also spent some of his time here in Australia at the Centenary Institute in Sydney with Patrick Bertolino, who, interestingly enough, was a postdoc at WEHI in the 1990s before establishing his lab up in New South Wales. So Jelte studied with Patrick looking at CD8 T-cell responses in the liver to different antigens. So it's clear that Jelte had developed an interest in liver stage malaria before coming here to work with us at WEHI. So Jelte's project in our lab has focused on the liver stage of Plasmodium falciparum infection, particularly undertaking functional genomic studies of proteins that we hypothesized are exported by the parasite into the hepatocyte to facilitate intracellular growth by the parasite and support its development. So this is a really challenging part of the life cycle to research due to lots of variability in many steps, but that includes the transmission stages, getting the gametocytes into the mosquitoes to start with, and then effectively using mosquitoes as a factory to produce, I would have guessed, Jelte, maybe millions of sporozoites over your PhD to actually conduct the sorts of experiments that you're going to present to us today. I was trying to add it up. I didn't know if it would be hundreds of thousands, but I suspect it's actually in the millions. And so that would be thousands of mosquitoes that Jelte has dissected to initiate his experiments. So as you'll see, Jelte has managed to do a substantial amount of work during his time with us at WEHI. Very fortunate to have him come to WEHI and work with us on this part of the life cycle. He's done substantial DNA cloning, transfections of P. falciparum, transmitted them through the life cycle and done both in vitro human hepatocyte infections with sporozoites, but also sporozoite infections of humanized mice with chimeric human livers. And that's acting as a model for human liver infection by the human malaria parasite. So I think Jelte's really pushed the envelope in terms of how he's developed a comparative analysis for us to measure multiple liver stage parasite knockout lines using humanized mice. And he's also revealed the complete lack of any utility of the RH3 pseudogene locus for expression of genes from that locus if you want to study sporozoites and liver stages. So we now know not to do that. And these are really important, obviously, as we move forward. So Jelte did all of this in a remarkably short time. I think might not have felt like it, but 3 years is a pretty short period of time to be doing these sorts of experiments, all whilst under, he's an international student here from the Netherlands in Melbourne during a COVID-19 pandemic. doing all of this through multiple lockdowns. Support from us, of course, but really a testament to Jelte's grit and determination to be able to finish his work in such a good amount of time. So Jelte, congratulations on completing your experiments for your thesis. I can say congrats on writing a great first draft of your PhD thesis. Especially since you're now back at home in the Netherlands and probably have many other distractions to keep you going, but you've done a great job so far. So good luck in completing that thesis writing, and good luck today in giving your seminar, which is also a necessary and important part of your PhD. So I'm getting a message here from Alan before we start that says this is not the correct place for Jelte's talk. So what are we seeing in the chat here, everybody? Can you see this? I think other people are coming in. We've got 26 attendees, so we're just going to, I think you and I are in the right place, Jelte, so we'll keep going. So yeah, without any further ado, Jelte, looking forward to your seminar today, and handing it over to you now. And I'll see you on the other side for some questions.
All right. Well, thank you very much, Justin, and thanks everyone for joining this seminar. It's a shame I can't see your faces, but nevertheless, I would like to talk to you about my PhD project involving the identification and characterization of exported hepatic effector candidates of Plasmodium falciparum. So Plasmodium falciparum is the causative parasite of malaria in humans. And even though there has been a very strong reduction in cases and mortalities over the past 20 years, mostly due to the implementation of insecticide-treated bat nets or antimalarials, it still remains a very big problem. And Plasmodium falciparum accounts for more than 97% of the deaths worldwide. So this indicates that it's the most lethal Plasmodium parasite that infects humans, and this is mostly in sub-Saharan Africa. So even though there has been a strong reduction in cases and mortalities, there are some problems, and especially the emergence of insecticide-resistant mosquitoes, the emergence of drug-resistant parasites, especially in frontline therapeutics in Africa as well as in Southeast Asia. But also the impact of COVID-19 has really disrupted the surface of specific antimalarials and treatments. So this is then visualized in this graph that you can see the case numbers increasing again from 2019 to 2021, probably due to the fact that COVID-19 had very strong impact in this disease as well. And this is also visualized in the mortalities in the red. So we can then interfere and continuously develop new therapeutics for malaria, and we can interfere in different parts of the life cycle. So the Plasmodium life cycle commences when an infected mosquito probes for blood and deposits sporozoites in the dermis of the host. And these sporozoites then travel to the bloodstream, finding their own way by traversing through different cell types. And these sporozoites then travel to the liver by the bloodstream. And within the liver, they again traverse multiple cells before they invade a single hepatocyte. And within this hepatocyte is formed this parasitophorous vacuole around itself. So this is the interface between the host and the parasite. And within this vacuole, it forms tens of thousands of merozoites that are in turn released into the bloodstream and start the asexual cycle of replication, also known as the blood stage. So there's also a proportion of parasites that are able to form sexual gametocytes, which are the sexual stages of the parasites and are taken up by the mosquito, and due to temperature shift, they are able to get fertilized and produce a zygote, and then an oocyte that travels to the midgut of the mosquito, in which it forms oocyst forms. So within these oocysts, then hundreds to thousands of sporozoites are formed and can be generated that travel to the salivary glands of the mosquito before they then invade a new host. So we can definitely interfere at different stages of the life cycle. But the interesting thing is that the liver stage is asymptomatic and therefore you can prevent disease, whereas the blood stage is the stage that causes the malaria-associated pathology. So what happens in the blood stage is that if you here see an infected red blood cell, you see all these knob-like structures, and that is due to protein export and the hijacking of the cell by the parasite. So if you then look more molecularly, you can see here the parasites in the bottom left, and you see that it is able to export all these proteins within the host red blood cell that have all these different subcellular localizations. So they make exomembranous structures such as Morris clefts. They present themselves on the surface of the red blood cell to interact with endothelial cells or with uninfected red blood cells, causing immune cell evasion and malaria-associated pathology. So what is at the basis of these proteins is usually amino acid motifs to target them for export into the host. So many classes of proteins, they have a signal peptide, which is an N-terminal hydrophobic domain, but it also has a Plasmodium export element, which is also known as a paxil, and this is defined by the pentameric amino acid motif, RXL, X, E, Q, or D. So if you have a simplified exported protein here, what happens to this protein is it is able to go into the endoplasmic reticulum of the parasite by its signal peptide. Then the paxil motif is able to be cleaved by this essential aspartyl protease called plasmapsin 5. So it's cleaved to conserve leucine, and the acetylated mature proteins are then able to travel to the parasitophorus vacuole lumen. So there's another class of proteins that are not processed but have transmembrane domains and do not contain a paxil. So these are paxil negative exported proteins, so known as PNAPs, and they sit in membranes of the parasite. So both classes of proteins are then able to be actively transported across the parasitophorus vacuole membrane by the PTEX translogon complex, which is comprised of 3 core components, namely EXP2, PTEX 150, and HSP 101. So for Plasmodium falciparum, it is estimated that more than 400 proteins are exported in the host red blood cell, having all these different subcellular localizations, such as Morris clefts, but also the presentation on the erythrocyte membrane causing virulence. So for falciparum, from these 400 proteins, approximately only 33 are conserved among other Plasmodium species. So this indicates that each Plasmodium species has its own export time, and you can't so always study Plasmodium falciparum using more easily accessible models. So that has been done in blood stage, right? But we are interested in liver stage because that is asymptomatic and we can prevent disease. So what happens in liver stage? So if you go into more detail, we have here a sporozoid entering the liver sinusoid, and due to very slow blood flow within the liver sinusoid, it is able to adhere to these heparan sulfate glycans. And it is also able to traverse multiple cells, such as Kupffer cells, hepatocytes, before they invade a single hepatocyte. And when they invade, they round up and they form this parasitophorus vacuole around itself. And within this vacuole, they then replicate into really tens of thousands of merozoites until they form a merosome that is able to be released in the bloodstream, which is the transition from late liver stage to blood stage. So this process for Plasmodium falciparum takes 7 days. For Plasmodium burgii, which is the species that infects rodents, it takes about 65 hours. So how is it possible that a parasite is able to develop within a nucleated host cell without the cell going into apoptosis or activating other cell death pathways, or without the immune system able to detect these cells? So we know that Plasmodium modulates hepatocytes, and most of these studies that have been done in the past use the more easily accessible model Plasmodium burgii due to the shorter life cycle of the liver stage. So what we know is that Plasmodium is able to recruit host cell Golgi, host cell ER, and host cell mitochondria. So they recruit the host cell organelles to be able to scavenge nutrients, and specifically, for example, lipoic acid. It is able to take up small molecules and metabolites to promote its growth, and also able to interact with apolipoproteins and other cholesterol markers. Then we know that UOS4, which is an essential player of the parasite PVM, is able to interact with endosomes and lysosomes. And we also know UOS3 is able to interact with autophagy regulators and thereby prevent clearance by autophagy. We know that the parasite is able to inhibit p53 and thereby inhibit cell cycle progression, or inhibit cell death, I mean, and promote cell survival by the promotion of the cellular inhibitors of apoptosis. So there's been a lot of studies been done in this space, and mostly in Plasmodium burgii. So what we then also know is that it has the same export machinery as blood stage. So plasmapsin 5, as well as PTEX components and pexyl-containing proteins are expressed in this stage. So is there any export being identified to date? Well, at least there is liver-specific protein 2, so LISP2, which is exported late during liver stage at 48 hours post-infection, which you can see in the host cell cytoplasm as well as the nucleus. And when you do knockouts of this exported protein, you see that the parasite has an abrogation in development of liver stages quite late. So more evidence arose when they did a proteomic study of merosomes. So this is the transition from late liver stage to blood stage, and where they identified that LISP2 is pexyl cleaved. So this indicates that LISP2 may be a substrate of plasmapsin 5 and is important for plasmapsin 5 mediated export, which is shown then in a study of our lab, is that when you then do a knockdown of plasmapsin 5 and you stain UOS4, which is the parasite membrane, the parasitophorus vacuole membrane, and then when you use LISP2 antibodies, you can indicate whether the LISP2 is exported across UOS4 or not. So when you then do wild type, you see nice exported LISP2. Whereas if you then do a knockdown of plasmapsin 5, you see that the export of LISP2 has been impaired due to the fact that they are probably not exported. So then when we do and generate mutants of plasmapsin 5 in Plasmodium burgii, we see that at 6 hours post-infection in the liver, we see that there's no difference in terms of parasites. But then in 24 hours, we see that the number of parasites has died considerably, and that's in 44 hours, that's the same. So basically, this shows that plasmapsin 5 in Plasmodium burgii is essential for early stage liver growth and survival. But this can't be attributed to the fact that LISP2 is not exported, or because it is not even expressed so early in liver stage. So there must be other substrates of plasmapsin 5 important for liver stage growth and survival. So this has been all done in Plasmodium burgii, but it is good to know whether there is conservation between the export machinery between Plasmodium species. So here we show that plasmapsin 5 and components of the PTEX translocation are also expressed in Plasmodium falciparum liver stages, which is the human parasite and more clinically relevant. So we show that the export machinery is conserved across Plasmodium species, which is more important. So to summarize that, we have shown for blood stage that there are more than 400 effector proteins being predicted and exported that cause virulence and malaria-associated pathology. Whereas for the liver stage, we have a very big question mark. We know that the machinery is expressed, and we know that there are pexyl-containing proteins, but within falciparum, there has been no protein been identified today that is located within the host cell that can be important for intracellular survival. So we hypothesized that Plasmodium falciparum exports proteins based on all the previous data and studies that we have shown during liver stage, and that is important for intracellular survival. And we have identified these three aims and basically divided it into three aims, which is the identification of these novel candidates using two different approaches, the expression analysis and localization of these identified candidates, and then the functional characterization using reverse genetics and the conditional knockout approaches. So we first focus on the first two. So we want to know all these question marks, right? Where do they go? Go they in the host? Go they in the nucleus of the cytoplasm of the host? And we wanted to know, we had two approaches. One was based on proteomics, but I'm not going to show that today. I'm going to show what we know about exported proteins, and that is paxil motifs. That's the only thing that we actually know that is related to protein export. So we have selected some criteria for selecting candidates that we want to look at. And so, for example, do the candidates have a paxil motif? And is the paxil conserved amongst other Plasmodium species, making it more likely that it is a real paxil motif? But also the presence of a signal peptide or any transmembrane domains is important, because that would target the proteins to the secretory pathway and therefore are more likely to be exported. But also, is there any evidence of liver stage expression? And because Plasmodium falciparum doesn't have any data on proteomics of liver stage or transcriptomics of liver stage because of the lack of very good models to study Plasmodium falciparum. So we have to base our evidence on proteomic studies from other species, such as Plasmodium burgii, like the merosome study I just showed you, or transcriptomics from Plasmodium burgii, to see whether there is an upregulation of specific proteins within liver stages. But also, another criteria that we are really interested in is the presence of a functional domain. And this would allow us to predict whether the protein has a function within the nucleated hepatocyte. Because the nucleated hepatocyte obviously has its own signaling transduction pathway, its own transcriptional regulation, and it is very metabolically active. So if you have a parasite protein and you were a parasite, then you would obviously try to interfere with these pathways and try to interfere with these host cell processes. And in a very related apicomplexan parasite, Toxoplasma, we know that many of these effector candidates have functional domains such as kinases or phosphatases to interfere with these host cellular processes. So these are just some examples of some criteria that we are looking for. So we generated a list of about 40 candidates, from which we then reduced to about 20. And we, amongst other candidates, we found out that we have a family of kinases. So we have here many candidates that were from the same family. So what is this family exactly? So this family is the FIC family of exporters kinases. And the interesting thing about this family is that there are 19 of these functional kinases in Plasmodium falciparum. All of them have a paxil motif, except FIC8. So some have been shown to be exported in the host cell. Some have been shown to be expressed in different stages of the life cycle based on proteomics. But obviously there's no data on liver stages. So no one has actually looked and see whether these FIC kinases are important for other stages of the life cycle. And the more interesting thing is that they are specific to the Leverania clade. So that means that all the other human infecting Plasmodium species, such as vivax, but also the rodent species, they only have one FIC kinase. And that is FIC8, which is the one that is not exported. So the only way to study this family is to actually do falciparum research. So what they've done in blood stages is that if you do a knockout of a specific FIC kinase individually, they see phosphorylating changes in the phosphoproteome of the parasite as well as the host protein, and then identified a whole network of what targets each FIC kinase have individually. So they see upregulation or downregulation of phosphorylation if you do a knockout of each FIC. So here they have identified this network, and some activate each other, some activate human proteins, and also some activate exported falciparum proteins. So each FIC kinase has its own targets. And we selected at least 3 of these FIC kinases to investigate further that I will talk to you about, which is FIC1, FIC3, and FIC10.2. So The interesting thing about these three is that there are three levels of expression. So FIG1 is slightly expressed in blood stages in quite low levels. FIG3 has not been shown to be expressed in blood stages, and FIG 10.2 is quite highly expressed. So we have three different levels of expression there. So what we can then do is we can look and tag these proteins and see localization and expression in liver stages downstream. So what we then have is we have this FIG kinase, and we generated these parasites by introducing this specific construct within the parasite. So we have a FIG specific domain here in blue, an intron, and then the kinase domain, which is the functional domain of these kinases. So if you get rid of the kinase domain, it basically is useless. And that had been tagged with an epitope tag, so 3HA, and we select with neomycin. So I've shown already before that 10.2 is quite highly expressed, and therefore also you can detect that using fluorescent microscopy, and you see that it is localized within the host red blood cell. But for us, we use usually a lot of flow cytometry for downstream liver stage assays. So we want to know whether we can detect these proteins also by using flow cytometry and HA antibodies. So we basically add these HA antibodies, and we see that for FIG1, we see a very slight shift towards the right here in the HA expression compared to wild type. For FIG3 HA, we don't see expression at all. And for FIG 10.2, we see a very strong shift. So that means that we can detect and use this as a proof of concept of that we can detect these candidates using flow cytometry in blood stages, which is important for downstream liver stage assays. So again, we are interested in liver stage. So obviously we then have to go all the way through the life cycle of the mosquito to be able to get these sporozoites and infect liver stage models. So what kind of models do we use in our lab? So the liver stage is quite long, and because if you want to see whether this protein is expressed in 7 of these days, it will take you probably years to actually investigate for each single protein. So what we can do is a very high throughput method, which is using HCO4 cells, which is the only known immortalized cell line that we know is able to be infected by Plasmodium falciparum. So that is good up until day 3. After that, this model can't be used and basically is not really provide any valuable information about infection of these cell lines. So then for later liver stages, we can use chimeric humanized mice. So these are mice immunocompromised with engrafted human hepatocytes. But these are extremely costly, so can't be used for every single experiment. So for the moment, I'm just going to focus now on the expression within HCO4 cells. And we do that by flow cytometry and HA expression. And then when we have a positive signal on a specific day, we can always look and see if we can localize it within the hepatocyte. So what we do is we have these HCO4 cells. We have sporozoites that are FIG1HA, FIG3HA, or FIG 10.2HA, and we infect these HCO4 cells individually with each FIG kinase. And we do that on day 0, and then on day 1, 2, and 3, we get the cells, we permeabilize them, and we fix them as well. And we then add specific antibodies, so HA antibodies to detect each FIG kinase specifically, but CSP antibodies, which is a protein that is expressed on sporozoites and liver stages. So that means that we can detect parasite material, which is important for the detection of viable parasites. So, and then we can use flow cytometry to be able to detect these cells. So here we see represented flow plots of, well, uninfected cells and then FIG1HA. And we see at 6 hours post-infection up until day 3, we see a nice shift in CSP positive HCO4 cells here, but obviously we can't see any HA expression besides some very small events. But that's not the same if you have an HA expressing HCO4 cell line as a positive control where you can clearly see that there is strong HA shift there as well. And we see that also for FIG3HA, indicating again a very nice CSP shift. So that means that we detect parasite material And for FIC 10.2 HA, we see that as well, even though we don't see any HA detection. So this probably indicates that on these 3 different time points, we don't see any expression of the specific FIC kinases that have been tagged with HA. But obviously, that has just been a snapshot of the life cycle. I mean, I just talked to you about that the liver stage is 7 days, and all the rest of that is about 1 month to get to that point. So that means that it's just a snapshot of the specific time, specific moments in time. And we don't know if these FIC kinases are important in the transmission to the mosquito or in the generation of sporozoites, etc. So the only reason and the only way to be able to know that is to actually look for it. And we can do that using functional characterization and reverse genetics, which means that you just knock out a gene and see if it has a defect anywhere else in the life cycle. So in Plasmodium, we transfect parasites within blood stages, but we don't know if we then have a defect in gametocytes or a defect in generation of oocysts within the mosquito or in liver stages, because that is where we are most interested in. So what we can do is we can use a conditional knockout approach, which is based on Cre recombinase in our case. So we express Cre recombinase within the parasite, and what happens is if you have gene X, so gene of interest, you can flank that by LOXP sites, which are identical sites and can be recognized by Cre recombinase. So after activation of this enzyme by adding rapamycin, this Cre recombinase then recombines the LOXP sites and basically leads to the excision, which is flanked by the LOXP sites. So this is a great way to be able to conditionally knock out a specific gene of interest. So this is what we have done with these FIC kinases. We have a FIC specific domain here in blue. Then we have a LOXP intron and the kinase domain. So what I mentioned is that is the functional domain. So if that is gone, it doesn't have a function anymore. It's just there, which has been tagged, and I've showed you that before. Then after that, we have another LOXP intron and GFP out of frame, and then a human DHFR cassette to select for resistant parasites. So if you add rapamycin, what I just showed you is that the region within the LOXP sites will be gone due to the recombination of these LOXP sites, leading to the excision of the functional domain. So that means that we have a mutant FIC kinase and a conditional knockout of the FIC kinase, which is now fused to GFP, and we can select using the resistance cassette for positively excised parasites. So how are we then going to determine whether we have successful excision is based on the amplification of these fragments. So in the full size, we have a fragment of about 3 to 3.5 kb, whereas in the knockout, we have about a fragment to 1.3 to 1.8 kb. So using PCR, you are easily able to detect after addition of rapamycin whether these whether the genome has excised or not, which you can see here that in PCR amplification, you see a clear size reduction, and that is also translated into a lack of HA signal for FIC1. So for FIC3, we also see a very strong reduction in size there in amplification, but and that has also been shown in FIC 10.2, where we also see a very strong anti-HA signal loss if we do a Western blot over that. So that means that we have successfully excised these FIC kinases during blood stages. So now we want to go to see whether we can form stage 5 gametocytes that we can then transmit to mosquitoes, and whether they form oocysts or salivary gland sporozoites, and then see whether they develop into late liver stages. So this process takes quite a long time. So we have to culture 17 days of gametocytes every day to make sure that we get to stage 5 gametocytes. Then we transmit those to mosquitoes, which takes another 16 days to get to salivary gland sporozoites, and then another 7 days of liver stage if you want to go all the way to the end. So this again means that we have a long way to go. And first of all, I would like to talk to you about the gametocytogenesis. So this is at day 17 of gametocytogenesis, and we then count the parasitemia of stage 5 gametocytes, where you can see that for Fig1 there is no difference between the stage 5 gametocytes, and the colors represent the same experiments. And here we see that as well for Fig3, that there's no difference, and for Fig 10.2 there's also a very similar amount of stage 5 gametocytes within the parasite. So that means that most likely these Fig kinases, so Fig1, 3 and 10.2, are not critical in the formation of stage 5 gametocytes in Plasmodium falciparum. So then we feed those infected red blood cells with the gametocytes to the mosquitoes. Then on day 7 to 10 we get the midguts of the mosquito. So we dissect the midguts of the mosquito, we stain them with mercurochrome to visualize the oocysts within the midgut. And then on day 15 to 17 we can look at the salivary gland sporozoites, because we dissect the salivary glands of the mosquito. So first of all, we go then to the oocysts. So here you can see the oocyst per mosquito, number of oocysts per mosquito, and each dot represents one mosquito midgut, with the number above the graph representing the mean of number of oocysts per mosquito. And the number below is the amount of mosquitoes dissected. So this is just one representative of 3 experiments, but you can clearly see there's no difference between the number of oocysts per mosquito. Then the other statistic that we can look at is the prevalence. So this means that the proportion of infected mosquitoes compared to uninfected mosquitoes. So infected means the mosquito has at least one oocyst within its midgut. But you can clearly see there's no difference for Fig1, and for Fig3 there's a slight decrease here, but that is a not significant difference in the number of oocysts per mosquito. And that is a similar trend that we see for Fig 10.2, indicating that these Fig kinases are not important in the transmission stage to oocysts within the mosquito. So that means that we can now look at salivary glands and see whether they can form sporozoites. So here at day 15 to 17, we have dissected the salivary glands of the mosquito and isolated the sporozoites. So here you can see the number of sporozoites per mosquito, and between these experiments there's a huge variation, but that's because we usually, within working with mosquitoes, you have a huge variation in the batches of mosquitoes, because they're still organisms, right? So here you can then see, if you color the same experiment, you see that there's no difference between the experiments and within the same experiment. For Fig1, for Fig3, and for Fig 10.2, indicating that, again, these kinases are not important for generation of sporozoites and the migration of these sporozoites to salivary glands. So if you then correlate that back to the number of oocysts, you can calculate the number of sporozoites per oocyst. And here we see that most of the numbers are between 700 and 1,000 per oocyst for Fig1 and Fig3, and for Fig 10.2 they're a little bit higher. But this again indicates that there's no difference between these parasite lines. So now we have sporozoites, and we can look at liver stage. So again, we have 2 specific models. The HF4 models are very good for early liver stages, and especially for sporozoite viability assays such as cells reversal or invasion up until day 3. Whereas chimeric humanized mice would be very useful for later liver stage to generate and see if the late liver stage development is impaired or not. So first go to hepatocyte reversal. So what does that mean exactly? Is that when a sporozoite moves through an hepatocyte, we use this molecule dextran fitzy that then is able to go into the hepatocyte and is light up the hepatocyte because of the invagination of the membrane. So then we can detect the percentage of dextran fitzy positive HF4 cells using flow cytometry. So here we have 6 hours post-infection that we see a slight increase in the number of traverse cells based on a Fig1 knockout. but that is not significant. And the same for FIC3, we see a slight decrease, but there is no significant difference, and that is a similar trend for FIC 10.2. So even though there's no significant difference, we see a trend among experiments for FIC 1 and FIC 10.2, indicating that it might be a biological difference. So the other statistic that we can then look at, another experiment that we can do, is hepatocyte invasion, which is based on the CSP antibody. So we detect the amount of parasite material that we have at specific time points. So here we see at 6 hours post-infection the percentage of CSP positive HF4 cells based on flow cytometry again. So here we see that again there's no difference between the different groups, such as the wild type FIC1 equivalent compared to its knockout. And that is what we see for FIC3 6 hours post-infection and for FIC 10.2 as well, even though there's a slight increase for FIC 10.2, but not significant. So this indicates that for 6 hours post-infection, there's no difference. And then we can do a time course for up until day 3, as I told you before. So on day 1, if you compare the grey graph on the left compared to its colored graph right next to it, you see that there is no difference at day 1 as well, or about 1 to 2% of CSP positive HF4 cells. Then the percentage will drop to at day 2, but the groups stay similar to each other. And in day 3, you really reach the limit of detection actually. So if you go later than day 3, you will definitely not get any useful information out of that, indicating that again, there's no difference up until day 3. So that has been the HA4 cell model and the in vitro model. But if you want to look at the late liver stage, we can use chimeric humanized mice. And because these mice are quite variable in the chimerism that they have generated, the way to get rid of this animal-to-animal variability is to co-infect these mice with 2 specific parasite lines. So this will reduce the variability, this will reduce the cost as well as the use of animals. So when you have done, traditionally, when you do a co-infection, you would have a wild-type parasite line and a knockout parasite line. So you would have wild-type compared to FIC1 knockout, for example. But we hypothesized that as long as you can distinguish these parasite lines, you can infect them with as many as you want, as long as it's practical and as long as you can distinguish them. So we hypothesized that we add all 4 parasite lines into the same animals. And this has never been done before. So we dissected all the mosquitoes at the same time, we counted all the sporozoites at the same time, and we then put them all in the tube and distribute them over 3 different humanized mice. And then on day 6, you can look for qPCR analysis or IFAs to be able to distinguish them. But we decided to go for a qPCR. So we have to look back into the actual genomic construct and see how these parasites look like. So here again, the blue domain is the FIC specific domain. So that means that we can design oligonucleotides specific for each knockout FIC kinase. And then GFP, we can design a generic primer to go the other way. So that would then have a 200 base pair fragment. Then the wild type, the problem that we have is if we use a normal wild type, so just a normal NF54 wild type background of this, we would not have a unique part of this wild type to be able to distinguish them between or against these other parasite knockout lines. So that means that we decided to go for the GFP luciferase parasite line. So this is a parasite line that has GFP within it fused to luciferase. So that means that the luciferase cassette is the only specific part of this parasite line, so you can distinguish the wild type from all the knockouts. So we designed oligos to amplify the luciferase cassette. So that's great, but we first have to actually validate whether our primers are specific enough. Otherwise, there's no point doing the experiment. We don't want aspecificity and cross-reaction of these primer pairs to other parasite lines. So what we then test in blood stages is that if you have here the FIC1 knockout primer pair, we can then use genomic DNA of each specific knockout. So we have FIC1 knockout, FIC3, FIC10.2 knockout, and luciferase, GFP luciferase, parasite line, genomic DNA. And then we use this specific primer pair in this case, and we see that only FIC1 knockout parasites have been amplified using this specific primer pair, which is great because that means that it's specific to the parasite line it should amplify. And that is what we actually observe for all the different parasite lines and different primer pairs. So we see that FIC3 knockout primer pairs only amplifies FIC3 knockout. FIC10.2 oligos only amplified FIC10.2 knockout parasites. And the luciferase cassette oligos only amplifies luciferase GFP luci parasite line. So this indicates that the primers are extremely specific and can be used in our assay. So what we eventually then do is infect these 3 mice with these 4 different parasite lines in the same animals, isolate the liver at day 6, so later liver stage, and then homogenize the liver, getting the genomic DNA of the liver, and do qPCR, so quantitative PCR, using these primer pairs. And here you can see the number of parasites per 1 million hepatocytes. So each dot, each colored dot represents the same animal. And here we can see that there's no significant difference in the amount of parasites on day 6. And the interesting thing is also that FIC1 knockout still has a wild type of FIC3, right? So that means that you can also compare them to each other because they're actually also their internal controls as well. So this means that there's no difference between these parasite lines compared to each other, indicating that these parasites develop normally into late liver stages based on the number of parasites. So to summarize all that, the FIC proteins, they lacked expression in parts of the liver stage infection that I've just showed you. And that is already novel results because no one has ever looked if these FIC kinases are important in other stages of the life cycle. So the conditional knockout was successfully established during blood stage. So actually it would be similar to traditional knockout, but by the addition of rapamycin, we could decide the moment that we want to excise it actually. So the FIC knockout parasites then did not show a defect in mosquito transmission, as well as in traversal and invasion of hepatocytes in vitro, and they developed into late liver stages at day 6 post-infection. So this indicates that all these parasite lines of the FIC kinases might not be important in the other stages of the life cycle, or they just are very compensating each other because it's a multi-gene family. So these are all possibilities. So then it remains me to thank all the people that are involved, and especially my supervisors, Justin and Ellen, for all the support throughout the PhD. Definitely the body labs, so past members as well as present members, and Robin and Brian for helping dissecting these mosquitoes for the humanized mice co-infections, because without you guys, I probably wouldn't have had these sporozoites anyway. So my committee members, Kelly and David, for the support as well. The Francis Crick Institute, so Mort Streaks Lab have collaborated with us on these FIC kinases and very generously sent these parasite lines to us. And then the project that I haven't even talked about, but Sepp and Kappa's group has also generated some material for us that we use for some of the experiments using another protein, VISP2, which I haven't talked about. But then I would really like to acknowledge Mel, because I think without an insectary facility, we would not be able to do any of these experiments. I think that she works incredibly hard by getting all the mosquitoes all the time ready for us to do our experiments. So I think without her and this facility, we would not do the experiments that we want to do. Obviously the ID2 division, because yeah, I have felt quite nicely home within the division for the past three and a half years. And I think it's also very important to acknowledge other facilities for WEHI, because I think we are extremely lucky at WEHI that we have all these facilities and all the infrastructure. So, you know, imaging facilities, fax facilities, histology facility, but also other facilities that do maybe not research related things such as media services. So they actually make our media for us. You know, I think it's very important to acknowledge that these people do work for us so we can do our research. So we are extremely lucky at WEHI, I think. So thank you very much for your attention, and happy to take any questions.
Thank you very much, Jelte. Roaring round of applause here and virtually for you. That was a very nice presentation. Considering it's now 7:53 in the morning,
Yeah.
you've done a tremendous job before breakfast. I think we can open it up to some questions. I'm looking in the Q&As at the moment. There aren't any, so I'd encourage people to have a think about a question for Jelte. I remind everybody that in Australia we don't do a PhD defence. This is it, right here, right now. This is your chance to test Jelte's knowledge. Maybe we could make him sweat a little bit. We don't really want to do that too much, but maybe just a little bit. We've got enough time for some questions. We have one comment. So well done. Congratulations, Jelte. This is from Eva in our lab. Congratulations, Jelte, to such a great summary of all your work. So well deserved. Are there any plans to look at liver stage egress with respect to knockouts? Yeah,
so obviously these thick knockouts, we have looked until day 6, and we obviously don't have the have decided to go to blood to or liver to blood stage transition, which is even later than day 6. So you would go to about day 7 to probably 10 to look at liver stage egress. But no, we haven't looked at that. That is obviously the only part of the life cycle that is still missing. So it might be involved in something related to that, but because we don't see any expression actually in this stage, I doubt that it might be having a function within this transition phase back to blood stages. We also see that in blood stages, they're not essential thick kinases, mostly probably due to the fact that they are a multi-gene family. They might compensate for the loss of one specific thick kinase. So yeah, that is obviously something that still can be looked at, but I would not be extremely confident that it would provide any information in terms of a defect to blood stage transition. Yeah.
Jelte, in the absence of other questions at the moment, I might continue along this line and ask you to describe a very challenging set of experiments, but I think they're possible, and I think they may have even been done in our lab. How might you go about addressing this particular question? So if you used your humanized mice, for example, how might you measure the downstream effect? And would you be able to do it with the multiple infections of the same mice that you've done, or would you need to do it singularly? Like how would you actually measure this?
So I think what you can do is you inject proportions of human blood cells into the mice at the time that the liver stage would be egressing. So the parasites have new human red blood cells because the mouse still has mouse red blood cells, so they can't invade mouse red blood cells. If you have human red blood cells, it might actually, they invade these human red blood cells. Then at a specific point in time, you can take all the blood from these mice and put them back into culture in vitro in a dish. And I think even with the 4 different parasite lines at the same time, because they're genetically different, you can still distinguish, doesn't matter what stage they are, you can still use the same primer pairs to actually look at the proportion of blood stage expression in specific conditions by qPCR. So I think there's no limit in the use of these primer pairs to specific stages of the life cycle. So yeah, I think that is usually what you would do.
Great. Great. Got another question for you, Jelte. This is from James M. I believe that's James Murphy. Well done, Jelte. How much sequence identity between the different Fic kinases? Is it surprising that there are different trends with the 3 you looked at, i.e. probably the blood stage expression? Or would you expect different substrates based on low sequence identity between the different Fics?
Yeah, so it's interesting because I think the kinase domain is extremely conserved among most of these big kinases, and most of the kinases anyway within the kinome of Plasmodium. So it is interesting that they find different targets, but they also identified similar targets that have been phosphorylated and activated by the same Fic kinase or by different Fic kinases. So that means that if you have one target, it can be activated by 4 different Fic kinases, for example. So yeah, some have different substrates, which may be because of the different localization of the proteins within the red blood cell. So this can be attributed to the domain that is non-conserved, that is maybe important for localization to specific compartments of the red blood cell. So for example, Fic 4.1 was localized on the membrane of the red blood cell, whereas Fic 10.2 was localized within the Morris cleft. So this can also then lead to the phosphorylation and activation of different targets. So I'm not sure if that is because of the different sequence homology between the actual Fic kinases, because the kinase domain is extremely conserved, but it might be because of the domain that is non-conserved that leads to different targeting or different localization, and that's why they have different targets. Yeah. Not sure if I answered the whole question there.
No, that's good, Jelte. We're at 5 o'clock. Wai Hong has snuck one more in there, and in the spirit of a PhD thesis defense, I'm going to allow it. Is there a pan-Fic small molecule inhibitor?
Well, that is difficult because obviously if you have a kinase inhibitor, it might also inhibit lots of other kinases that you maybe don't want. So I think it might have, I don't think there is a Fic inhibitor today. So it can be very interesting to make that. So then you can actually knock out each Fic all at the same time, which would allow for more interesting results, I think, to see if there is an importance of this whole family. But I think it might be hard to target specific Fic kinases individually with an inhibitor, but also it might also be that these inhibitors might interfere with other kinases that are important for Plasmodium development. And you don't want a specific effects of other kinases, and then you think, oh, well, this can be because of Fic kinases, but it is important to then develop a very specific inhibitor. And it might be possible, but I'm not sure how you would design that, to be honest. Yeah.
Alan Cowman has sent one through. It would probably be remiss to not read that out from the Deputy Director and also your co-supervisor. Congratulations, Jelte, and I love the way he says and spells your name, Y-E-L-T-A. Congratulations, Jelte. I think there's 1 or 2 Fics in Bergii. Have they been knocked out, and are they essential in blood stages?
Yeah, so Bergii has Fic 8, and that's conserved among all Plasmodium species, and there's a study in liver stages that have actually knocked out this Fic 8 and seems to be important for liver stage development. It's also important for blood stage growth in falciparum. So this Fic 8, and probably due to evolutionary development, might be the one that is actually important for the parasite within the essentiality of the parasite development throughout the life cycle. So yes, there has been Fic 8, but yeah, that's not exported. So it's for us less interesting, I guess. But yeah, definitely it might be worth looking in liver stage to see if FIG8 is important in falciparum development. It might probably not localize to the host cell, but still it can be interesting to look at. Yeah.
What if it was a PNEP? Maybe it's translocated to the host without a PEXL. So there's this other class of proteins, as you mentioned in your talk. Maybe it is a PNEP. So I think there's.
But I think in blood stages they have shown that it is not exported. Yeah. Doesn't mean that it is not exported in liver stages, but it might be unlikely.
So that touches on a final note that Wai Hong had mentioned about are there any essential FIC in any life cycle stage, and I think you've covered that. So thanks very much everyone for your questions. Really appreciate them. Jelte, it's a touch after 8 a.m. I think you can go and have a Dutch breakfast. A Dutch breakfast is 4 mils of Galliano, 30 mils of lemon juice, 45 mils of Advocat, and 45 mils of dry gin. Congratulations. Thank you so much for a great presentation and for choosing our lab to come and spend 3 years of your life. It's been great, and look forward to finishing up your thesis and publishing this work too. Thanks a lot.
All right. Yeah, thank you very much.